experiment where we compare the effect of prenatal stress in normal mice as well as mice with a genetic variation. In this case, the serotonin transporter. That's the transporter responsible also for the reuptake of serotonin, also the target for antidepressants like Prozac. I'm not going to show you those data. They are still being analyzed at the moment. I will show you an ongoing research line within our lab, and actually I think last year 2 junior students of your track worked on that project, and one of them is now doing a senior internship on this project. And actually a previous student now is, and he's hopefully in January, depending on Steve, on this line of work. So getting back to the scheme of what we do, now focusing on aging and Alzheimer's disease, see to which extent Alzheimer's disease involves epigenetic regulation or dysregulation. So this is Leonis Kouliaris, our Greek PhD student on this project that will finish, who will finish soon. Of course, he left Greece for certain reasons, not being financial, but of course he's not going back at this stage. And he did a hell of a job here in our group. And gathered a lot of information and publications. He's now doing a clinical training in England. Also, one of the junior students was also. Anyway, so first, what he started to do, there was not that much known about epigenetics in Alzheimer's disease or in disorders, except for cancer, of course, again. And he first started to collect evidence. I wrote a nice review on that in 2010, and then he got really back to work to do some experiments by himself. He also showed that gene-environment interactions are pretty important in Alzheimer's disease, which is also rather a new concept. So one of the big studies that he did, or actually he had data from that study, a brain tissue available. It's quite a unique study because you don't regularly have access to material like this. So what was the cohort based on? It's the Maastricht aging study, the Maastricht mouse aging study. We have animals of 12 months of age and 24 months of age. So if you know that an animal, a mouse is already adult at 3 months of age, we're dealing with really old mice. 12 is already really old, and 24 is really. You have different groups. Either they were exposed to a normal diet, which means they have 85% of what an animal normally eats when fed ad lib. And ad lib means that they have unlimited access to food and water. They then also, of course, eat too much. So to control for their intake, we gave them 85% so that we know that they actually eat everything they get. And in the caloric restriction group, they got 50% of those calories. And it seems an awful low amount, but actually it has very positive effects on things like lifespan and also the quality of life. And this is not just something known from this experiment, but also from other experiments and also from human data. So 2 different conditions, and actually they also included genetic variation. So we have animals that showed different genetic background in terms of superoxide dismutase and antioxidant. The idea was if they overexpress human SOD, this would protect them from aging effects. You can forget most of this because we didn't see that many effects of this genetic variation. So particularly we see effects of aging itself, comparing 24 versus 12 months of age, and then effects of this protective caloric restriction. Then a second piece of work, which I will also show you, he compared normal mice, wild-type mice, with animals that are genetically modified to develop plaques in the brain. So these amyloid plaques are readily seen in people with Alzheimer's disease. And then he compared different ages. In those animals, 9 months is already really old because they develop plaques and show cognitive impairment already from 6 months of age. The first study, again, I said, these are different groups, and what he did, he investigated the effects of aging on epigenetic measures, and he of course looked at the effects of the genetic variation as well as the environmental variation. This is some basic data, survival curves. So here you see the 2 age groups that we will discuss are here and here. But what you see here is that later on you see a clear distinction in the survival curves. Some animals already start dying here after they reach 1.5 years of age. And you see a clear difference here at this stage where the animals that were exposed to caloric restriction, so these are the two upper groups, much more of them still are alive at that time. Here there was an infection into the animal facilities and all animals died. And we couldn't use the brain. Fortunately, we just sacrificed them several groups at 24 months of age. Such an aging study is almost impossible now because I think the last 3 years there have been 3 big rotor infections and then you need to sacrifice all animals. Anyway, in this case we were lucky. And what you see here then is that caloric restriction already expands life span. So it extends your the age that you reach. In this case in the mind, but this has been shown also human data as well. I'm not saying you should not eat or that you should do something like anorexia. No, there's somewhere there's a balance. Of course, we eat too much. You also see difference in weight, of course. The caloric restrict animals are of course life, but this can be expected. But it's a big difference. And what we then did, we particularly are interested in the hippocampus, because this is a region that is critically involved in emotional regulation, but particularly also cognitive behavior, so learning and memory. And it is also known to be critically effective in Alzheimer's disease. So that's why we focus here on the hippocampus. And this is a slice of a hippocampus of a mouse. In this case, stained for a certain epigenetic marker, but I will show more on that in the coming slide. But this is how the hippocampus looks like, and you might recognize this region. It's the dentate gyrus, and this is a region where we also still see formation of new neurons in adults. So what was previously thought is that the brain is formed at birth and it can grow a bit, but there are no new cells are being formed. It can just go down in quality and quantity, and that's it. Well, it has been shown that there's 2 brain regions where you still see proliferation of nerve cells in adulthood. And one of the 2 is the hippocampus. And this neurogenesis, as it's called, is also considered to be a mechanism for learning and memory processes. And with aging, you still see some decay in neurogenesis, and this may be responsible for the fact that you remember less, etc. And it's difficult to form new memories. So the hippocampus is a very important region. And he investigated different parts of the hippocampus. They all have differential function, not important for now. So this is DMT3 immunoreactivity. I told you you can do some epigenetic related experiments by just staining for a player that is important in epigenetic regulation. So in this case for DMT3A, which is one of the DNA methyl transferase. Also very important in these dividing cells, and these dividing cells are darkly stained for DMT3A. You see here the general immunoreactivity pattern, so almost all cells in the hippocampus are stained, but some cells, if you go on these higher magnifications, are stained much more than others. And these are the cells that recently divided. Because if you have division of a cell, you need to duplicate the DNA, but you also need to duplicate the methylation patterns. And that's why you see very high levels of these DNPs. So we see here dividing cells, or cells that have recently undergone cell division, and we see, let's say, differentiated cells, which also still show DMT3A activity. So we just use an antibody against DMT3. Then he compared the different groups, so complicated in terms of the load of information. What we always see is that with aging, in black you always see the old animals, in white, well, can't say young, but in this case it's a younger group. And what we always see is an increase with aging, at least in the animals that were given a normal diet. The group on the right here were the animals that were exposed to this caloric restriction. So what we see here in general is an increase in the levels of this enzyme, of this DMT3A, with aging in normal animals, but when they are exposed to caloric restriction, we don't see this aging effect. We see that in different regions of the different animals, it's always the same effect. So there's age-time diet interactions and an increase of DMT3A with aging, which can be prevented or attenuated with caloric restriction. Whether this connects to function, you don't know yet, but it's an observation. And these were levels of immunoreactivity for these differentiated cells. Then he also looked at these dividing cells, did a co-localization PRU, so that is something that is built into the DNA when cells are duplicating, and he saw co-localization. So all these cells had recently undergone proliferation as a proof of principle. And what you see here, it's only an age effect, meaning that with aging you see less of these cells. There was no protective effect of the caloric restriction, although the power was also a little bit low. In all cases, there's no effect of this gene effect, so you can ignore that on purpose. He also looked at heterochromatin versus pure chromatin, so compact DNA versus non-compact DNA, and this confirmed the findings that this high level of methylation is also associated with more compact methylation. Then he did the same for methylation itself. So of course we have an enzyme that is causing the methylation, but you can also use an antibody against methylation itself, so 5-methyl C, 5-methyl cyanidine. So this is the cyanidine that's being methylated in a CpG unit, C followed by G. And you see the similar pattern with an increase in methylation with aging, but it nicely fits the pattern of DNP3A, and this effect is not seen in animals that were exposed to growth restriction. This growth restriction is really for meaning almost, so from 4 weeks of life until the 2 years of age. This project, just for the animal cost, I think is more than 200,000 euros. So that fits, and then it was recently discovered that there is another type of epigenetic modification. Was this discussed during the course already? Hydroxymethylation? Yeah. No. So until a few years ago, they knew the enzymes that were responsible for methylation. So here we have an unmethylated cyanidine. This is converted into a methylated cyanidine by these DNP, so one of them is DNP3A. So that's the active process of methylation. They, however, did not know how the methylation signal could be removed again, because you don't want this to persist forever. In certain cases, you want to regulate it and change again gene expression by taking away this method. And then they discovered the process of hydroxymethylation. So there's a certain enzyme called TAP that converts a methylation signal into hydroxymethylation. Apparently, two things happen. One is that eventually this hydroxymethylated C can be converted again into a C, so you can lose the methylation signal. Another thing is that this hydroxymethylation changes the affinity of your cytosine with certain transcription factors and methyl binding domain proteins. You have heard of methyl domain binding proteins? These are proteins that bind to methyl groups in the DNA and then actually recruit these players that cause chromatin compaction. So this also links methylation to chromatin state. For example, these H-tag compounds, they bind to methyl binding domain proteins. One of them is MECP2, so MECP2, and they then eventually cause chromatin compaction. If you have an hydroxymethyl C, so an hydroxymethylation at this cyanidine, this changes the affinity, and eventually, because you lack the histone deacetylases, you get a more open state of the chromatin. Again, this is a rule. It doesn't mean that in all cases this is the way it works. So this is the process of hydroxymethylation. So what we did is we also quantified hydroxymethylation. And the important thing, and that's something I should mention, maybe somebody has discussed bisulfite treatment technique, no? So the way to detect methylation at a gene level, and if we look at every single CpG in a gene to see whether it's methylated or not, that's bisulfite treatment or bisulfite sequencing or bisulfite-based arrays or whatsoever. And I think there have been several dozens of papers in big journals like Nature and Science who either reported differences in methylation between two conditions or actually did report no differences, and they were based on this technique. Now it appears that this technique cannot discriminate between methylation and hydroxymethylation, so it gives a signal for both, and you don't know whether this was methylation or hydroxymethylation. Actually, this means, considering the opposite effects of methylation and hydroxymethylation on gene expression, that if you find a difference, you don't know whether it has a positive or negative effect on gene expression because you don't know whether it's methylation or hydroxymethylation. And more importantly, if you don't find a difference between your two groups, this may still be because they two balance each other out. What I mean with that is that there have been several papers in high-impact journals, of which we now don't know what to think of it. We don't know to which extent we can extrapolate the findings. And this was also the case for these GR findings that I showed you in the beginning. So a lot of these effects of internal behavior on the expression of the glucocorticoid receptor have never been replicated, purely because of the fact that now apparently this was not methylation but hydroxymethylation. So techniques are quite important. And challenges are reported in the paper that I used to show you that we recently published in all kind of research. hydroxymethylation, but methylation. This is the same cell. You see that different parts of the cell are actually, different parts of the chromatin are showing high signals for either methylation or hydroxymethylation. Already showing that they are associated with different states of the chromatin. And what he also showed then, again, of course, in the same set of animals, the effects with aging and the effects of flow restriction. Actually, he shows the same, that with aging there's also more hydroxymethylation. Whereas this effect is not seen when you expose animals to flow. So you still don't know exactly what is the balance between methylation and hydroxymethylation and how does this affect gene expression. Very importantly, these are global measures. You don't talk about any specific gene here, and that's of course the next step. You see different effects of aging, of these diets, but what does this do at a single gene level, and which genes are more affected by these changes than others? That's something we are currently trying to find out. We also looked at chromatin players like HDAC2. You can also just simply use an antibody against HDAC2. And why is HDAC2 important? Well, that one has particularly been shown to be important in relation to Alzheimer's disease. Just as well as HDAC5. Aging inhibitors, as said before, used in cancer therapy, but they also have shown to improve learning and memory processes in different models, in wild-type mice, but also in Alzheimer's mice. And it was also shown that this directly changes pathophysiology. So the normal pathological hallmarks that you see in Alzheimer's disease, and in this case in these Alzheimer's mice, they are either decreased or prevented by treatment with HDAC inhibitors. The problem again is what are the side effects? Because we have also tested HDAC inhibitor in the lab, and these animals get sick to a certain extent. So the question is, could you actually use such a treatment in the long run to try to cure or prevent Alzheimer's disease? I would say no at this stage, but we try to get it more specific so that it doesn't affect also genes involved in cancer. And so what we did here, we saw again similar effects, increasing effect with aging, so all more or less the same, which is prevented by caloric restriction, suggesting that in general epigenetic variation is different with aging, or it's affected by aging. We see increase in DMP3A, methylation, hydroxymethylation, and HDAC2 with aging. And these effects can be prevented by environmental variation, in this case caloric restriction, but not by this genetic modification. And then the next question is, of course, which genes are we affected? So that's currently under investigation. But these were all wild-type animals, or we have this modification in superoxide dismutase. We, of course, now extended this to Alzheimer's mice to see whether this is sort of accelerated form of aging in terms of epigenetic processes, or whether something else is going on. So the overview of the study design, we have young animals. This is when blood formation starts in these affected animals. And we have animals that were relatively old, between 9 months for Alzheimer's mice, and the mouse was already quite old. So we investigate the effect of aging again, in this case genetic variation, and these genes that are important for Alzheimer's disease. And actually we also did prenatal stress in an Alzheimer's model. So we stressed a pregnant woman, and then look whether her offspring, in this case Alzheimer's mice, showed cognitive impairment early in life, and they did. So in this case it's a combination of things that you saw in the first part, developmental stress in an Alzheimer's mouse model, and then you see actually an acceleration of cognitive decline. I will not show data on that. I simplified the schemes here a bit, but what we see in general is that in wild-type mice we again see this aging effect. So this is methylation. We see this aging effect that we saw before, and actually more striking during earlier ages, because you saw the other mice were 12 and 24 months of age, and the difference was not that big, although it was significant. It wasn't huge. In these animals from 9 and 3 months of age, the difference was much bigger. However, there's something completely else going on in these Alzheimer's mice. They don't show an age-dependent effect, if at all, it goes in the other direction. Interestingly, the levels here are more or less comparable to wild-type levels at an early age, so it doesn't increase. Here, actually, you see for hydroxymethyl C that the levels in the Alzheimer's mice are already high at the start, so they match actually the old group. And what this suggests is that if you consider the fact that they have opposite effects on gene expression, is that there is a disbalance when compared to wild-type animals between methylation and hydroxymethylation, because the methylation is similar to the young animals, whereas the hydroxymethylation is already similar to the old animals, the normal old animals. So So there's a disturbed balance between the two. And what's very important to see, to conclude, is that it's, for sure it's not a sort of accelerated aging, because then you would expect here that the levels would be very high, even increase much more with aging, and they don't. So apparently there's something else going on in Alzheimer's disease. And that's something you can see here as well. These were brains from a collaborative group in Sun City in Arizona. It's the Banasson Health Research Institute, and actually people are there in a sort of a dorm room or elderly facility. I'm not saying these people are waiting for them to die, but as soon as they die at least, then the brain is immediately safe for research purposes. And actually, these people are tested at least once a year for all kinds of cognitive scores. So these people, we know everything of these people. We know how they function cognitively, we know blood load, we know tau load, tangle load, we know almost everything of these subjects. With this group, we last year started a collaboration. This is what they published already